name is Alexandria Huang, and I am currently an intern at the Grin Staff Lab here at BU researching CT scanning. The cells of various diseases of the world possess different types and frequencies of specific proteins that help to differentiate them from one another. Our research project looks to utilize microcomputed tomography, or micro-CT, to create 3D reconstructions of tissue samples and image specific proteins within them to allow for effective diagnosis of diseases. Almost like a flashlight, micro-CT allows us to see the shadows of a targeted protein within cells by shooting the cells with X-ray beams, and allows us to visualize the location and frequency of those specific proteins, which in turn could help us to find vital information about the type of disease a patient is suffering from. Our focus during research was to find a specific method to make those targeted proteins visible to micro-CT. We hypothesized that by using brominated or iodinated nucleotides, we could find an effective method for making these proteins visible to X-ray beams, as both brominated and iodinated nucleotides absorb the beams of X-rays. We experimented with annealing antibodies that bind to the specific protein we are trying to find in a cell to a rolling circle amplification template, or RCA template. The template would then be attached to a target protein and create a long mass of nucleotides to form a matrix. Micro-CT could be an extremely beneficial method of creating 3D reconstructions of models of tissue and enable for diagnosis of diseases based on the presence and frequencies of those proteins. For example, there are various types of different cancers. So if you're to take perhaps a breast cancer cell and a liver cancer cell and use micro-CT to compare them, you would find that perhaps the breast cancer cell had a higher frequency of a certain protein as in comparison to the liver cancer cell. Another application could be patients with liver problems could go to the doctor, and the doctor could use pre-existing methods of imaging to determine that the liver was abnormally large. That's not as detailed as micro-CT, and with the help of micro-CT, a doctor could take a sample of the liver cells, scan the sample using micro-CT, and then diagnose that the patient had non-alcoholic fatty liver disease based on the distribution and abundance of specific proteins within that cell that are associated with fibrosis. As of now, micro-CT could play a vital role in helping properly diagnose various patient diseases and help treat them as soon and as effectively as possible.